Hello students, welcome to EPG Pathshala. I am Dr. Jiten Vasist from Department of Biotechnology and Bioinformatics, JP University of Information Technology, and today I am going to tell you about the module that's nucleosome packaging. This module comes under paper Cellular and Molecular Biophysics. Nucleus is the central part of a eukaryotic cell, which is approximately 10 micrometer in diameter, and this tiny organelle has to accommodate a length of linear DNA of approximately 2 meters of length. Present modules deals with the packaging of the linear DNA with different proteins so that it gets accumulated in the nucleus, and therefore present module focuses on the following learning objectives. Number 1, to elucidate the structural organization of a nuclear assembly. Number 2, to understand the basic concept of DNA packaging in nucleus with reference to binding capacity with different proteins. And number 3, to classify different architectural proteins on the basis of their structural and location specific features in a nucleosome. And lastly, number 4, to elucidate the effect of nucleosome packaging in gene regulation processes. The human haploid cell contains 23 chromosomes and approximately 3 billion base pairs of DNA are present in it. As majority of human cells except female ova and male sperms are diploid with 23 pairs of chromosomes, they contain a total of 6 billion base pairs of DNA per cell. Due to these high number of DNA base pairs, and as rise per base pair is around 34 nanometer long, each diploid cell contains about 2 meters of DNA. You can get calculation of this, and these calculations more get complicated when an estimation of total number of cell is done. The human body contains approximately 50 trillion cells and therefore contain 100 trillion meter of DNA per human. Now consider the fact that the sun is approximately 150 billion meters from the earth. Thus, each of us has an adequate amount of DNA to go from earth to sun and back more than 300 times. 2 meters of DNA strand in a single diploid human cell must coil and twist over many times in order to fit into the cell's nucleus, which is about 1/10 of the human hair in diameter. However, this problem gets sorted by virtue of structural features of DNA molecules which make it a negatively charged moiety due to the phosphate backbone. The negatively charged DNA makes a complex with a group of positively charged proteins, and these structures are termed as chromatin. Figure 1 shows the chromatin structures. Highly condensed structure of chromatin ultimately forms chromosomes. Eukaryotic chromatin under a microscope resembles like a bead on a string in which DNA is wrapped around nuclear protein, and this organization forms the fundamental repeating units, which are basically known as nucleosomes. Nucleosomes are seen in figure number 1. Although nucleosomes were previously observed as particles using electron microscope by Dawn and Ollins in 1974, but their structures were lastly elucidated by Roger Korenberg. Now, if you see, there is a group of proteins which is known as histones. These histones are basically positively charged proteins which associate with DNA in a nucleosome. These positively charged histones pack together with negatively charged DNA into a microscopic space of eukaryotic cell nucleus and takes part in the formation of chromosomes. The coiling of linear DNA requires energy, and within the nucleus, histones provide energy in the form of electrostatic interaction to fold DNA. This results in chromatin, which can be packed into much smaller volume than DNA alone. Histones are generally present in the nucleus of eukaryotes, archaea, however absent in bacteria. In eukaryotic cells, 5 different types of histones are present at specific locations, and among these 4 histones H2A, H2B, 3 and 4 are found in the core, while histone H1 are present as the linker histone at outside the core of the nucleosome. All core histones are present in equal amount, whereas H1, which is a linker, is about half of the other histones. Histone H1 is also known as linker histone as they bind to linker DNA during nucleosome assembly. If you see in the figure, it shows that it is a family of 126 different members from different species, and members of this family varied in molecular weight of about 21 to 29 kilodalton. The binding of H1 with linker DNA is responsible for compaction of nucleosome. Hence, based on their association, they have been classified into stronger, intermediate and weak chromatin condensers. In humans, there are about 10 histones H1 variant has been reported so far. This figure shows the schematic representation of organization of different histones with DNA in a nucleosome. Now we'll talk about histone H2A. Histone H2A is a part of core histone octamer of a nucleosome and form dimer with H2B. Most of the protein of this family contains approximately 14 kilodalton protein consisting of approximately 130 amino acid residues. Histone H2A is a family of 265 different members from different species. Among histone core, H2A have maximum number, approximately 19 invariants. H2A undergoes many post-translation modification like acylations, methylations, citylation, phosphorylation, etc. If you see the other histone that we call the H2B from dimer, it forms a dimer with H2A and is also a part of the core histone of octamer. Most proteins of this family contain approximately 14 kD and consisting of approximately 126 amino acids. 19 variants of H2B have been reported in humans. Only few translation modifications of H2B are known yet. It seems that PTMs are still not studied well in H2B. Another histone that we call the histone 3 forms a heterotetramer with H4 and form core component of histone octamer in nucleosome assembly. They are about 15 to 16 kD with approximately 135 amino acids long sequences. H3 is a family of 216 different members from different species. H3 has 6 different variants in humans. CNPA variant of H3 is bigger in size and exclusively incorporated into centromeric regions of nucleosomes. Other variants are supposed to provide damage memory as they are incorporated in sites of DNA damage. Number of post-translation modification at some conserved regions of H3 control major biological functions. Functions of some modification has been mentioned in table 1. If you see the table 1, we will find the modification and their utilization. If you see that methylation at lysine 4, it activates the transcription, while methylation in lysine 9 and lysine 27, it represses the transcription repression. Now we'll talk about histone 4. Histone 4 along with histone 3 forms a heterotetramer and constitutes the nucleosome assembly. There are about 11.3 kd proteins consisting of 102 amino acids. Histone 4 is a family of 116 different members from diverse species. Single H4 has been reported in humans. Among all histones, H4 is most conserved and also acts as a docking site for other histones. Post-translation modification such as phosphorylation, acetylation and methylation at H4 involves in controlling transcriptional activation, DNA damage as well as cancer hallmarks. As histones are most important for DNA packaging into chromosomes, they are highly conserved among eukaryotes. Sequence of H4 from a pea and cow differ at 2 out of 102 positions, and this is the best example to define evolutionary conservation of these proteins. Hence, any change in histone sequence will be lethal to a cell. Now we'll talk about DNA length in a nucleosome. The amount of DNA per nucleosome was determined by treating chromatin with DNase enzyme. Micrococcal nuclease is one of the such enzyme, has an important property of preferentially cutting the linker DNA between a nucleosome prior to the DNA, that is wrapped around an octamer. If aminase treated chromatin DNA is separated on gel, number of bands will appear, each having a length that is multiple of mononucleosomal DNA. Nucleosome is a basic repeating structural and functional unit of chromatin, which is composed of 146 base pair of DNA around 8 histone proteins. Nucleosomes are assembled structures of core histone octamer composed of each histone 2A, 2B, 3 and 4 together, and wraps approximately 1.7 tons of 146 base pair DNA. This nucleosome structure will appear as string on beads under microscope. The linker histone H1 wraps another 20 base pair, resulting in 2 full turns around an octamer, and this structure is called as a chromatosome. Now we'll show some of the histone characteristics for nucleosome assembly. Histone sequence during packaging are highly conserved. They contain high content of positively charged amino acids, hence close association with negatively charged DNA. Disc-shaped protein core of nucleosome is assembled in ordered only in association with DNA. Otherwise, histone core will form intermediate assemblies in solution. All core histones have a histone fold domain that mediates assembly of histones, only intermediates. Nucleosome assemblies also involve ordered assembly of histone with DNA. Histone folds mediate the formation of heterodimers of specific pairs of histones. Firstly, H3 and H4 forms a heterodimer, and then they associate with 2 molecules of H3 and H4 to form a tetramer. This is seen in figure number 3. Quite the reverse. H2A and H2B forms only heterodimer in solution but not tetramer. This figure 3 shows assembly of different histones to form a nucleosome. The histone and a DNA interface in each nucleosome is extensive. It contains 142 hydrogen bonds between DNA and histone core. DNA-histone interaction is not fixed. Therefore, dynamic interaction responsible to maintain these structures is needed. Structure of DNA is wrapped in a way that allows the binding sites of regulatory protein is in accessible linker region of DNA. Most of the bonds are found between phosphodiester backbone of the DNA and amino acid backbone of the histone proteins. Apart from hydrogen bonding, hydrophobic interactions and salt linkages are also responsible to hold nucleosome structure as a stable one. For example, in each histone octamer, lysine and arginine are about 1/5 of all amino acids. Positive charge of these amino acids effectively neutralizes negative charge of DNA backbone, which stabilizes the structure. In addition to histone fold domain, extended end terminal of each histone is called a tail, as it lacks any defined structure and they extends out from the histone DNA core. This end of DNA histone is more flexible and able to interact directly with DNA and the different proteins within the nucleus. There are several covalent modifications at histone tails which are responsible to control chromatin structure and function. In nucleosome positioning, specific regions of DNA have higher affinity for nucleosome. For example, minor groove of AT-rich region DNA faces core octamer of histones, whereas minor groove of GC-rich DNA have opposite tendency. Alternating AT and GC-rich sequences are rare in genomic DNAs. Now we will talk about a higher order structures of chromatin. The packaging of DNA into nucleosome shortens the fiber length about 7-fold. It means 1 meter piece of DNA will become just 14 centimeter long. Although this shortening approximately half foot of chromatin is still too long to fit into the nucleus, which is approximately 10 micron in diameter. Consequently, chromatin is further have to coil into an even shorter and thicker fiber, that's we call the 30 nanometer fiber or solenoid, because it is about 30 nanometer in diameter. Over many years, there was a great deal of assumption concerning the matter in which nucleosomes are folded into 30 nanometer fibers. This 30 nanometer fiber may be highly irregular. Interestingly, H1 is the most important in stabilizing higher order chromatin structures. Different higher order packaging are seen when these solenoid further condense into form a chromosome. This figure 4 shows an overview of different levels of folding of DNA into chromatin structure, and each level will show you the highly condensed structures. If you see the dynamics of chromatin structure, you will find that the chromatin structure not just serve as a way to compact DNA within the nucleus, but also provide coordination for unfolding the bound DNA for many gene regulation pathways. In eukaryotic cells, some genes are not expressed unless their genes accessed by RNA polymerases and transcription factors to translate into mRNA and finally into a protein. In its packed chromatin state, the tight coiling limits the access of these substances to eukaryotic DNA. Therefore, in order for gene expression to take place in a cell chromatin, it must open and this process of opening is we generally call it chromatin remodeling. The chromatin remodeling is of vital importance for proper functioning of all eukaryotic cell. Various chromatin remodelers provide mechanism for chromatin modification and allowing transcription signals to reach their destination on the DNA strands. Investigations about chromatin remodelers reveal they are large multi-protein complexes that utilizes the energy of ATP hydrolysis to assemble and reform nucleosome. Recall that nucleosome wrap 146 base pairs of DNA in approximately 1.7 turns around a histone octamer disk, and the DNA inside each nucleosome is generally inaccessible to DNA binding factors. Remodelers are therefore vital to provide the access to the underlying DNA to enable transcription, DNA repair, chromatin assembly, and other processes. There are a number of ATP dependent chromatin remodeling complexes, and function of each complex is controlled by the cell. ATP hydrolysis and remodeling complexes catalyze nucleosome sliding and make other proteins to access to nucleosome DNA. These changes occur in three forms. Number 1, sliding of histone octamer. Number 2, transfer of histone octamer from DNA. And number 3, remodeling of nucleosome to increase the access of DNA. Some negatively charged histone chaperones and remodeling complex remove or exchange nucleosome core. Presence of specific type of remodeling proteins is responsible to maintain chromatin dynamics. For example, some proteins allow the formation of nucleosome, whereas other may create obstacle for the same. Role of histone modification in controlling dynamic and chromatin structures. Then you will find that variety of covalent modifications such as phosphorylation at serine, methylation at lysine, and mono, di or triacetylation at lysine occur in a side chain of core histone of a nucleosome. Among all these number of modifications occur at 8 unstructured and terminal histone tails. All these modifications are reversible and look out by specific enzymes. Each side chain is modified and removed by specific enzyme. For example, histone acetyl transferase, that's we call the head, is responsible for acetylation at lysine, whereas histone deacetylase complexes, that's we call the SDACs, remove acetyl group. Similarly, histone methyl transferase responsible for addition of methyl group to lysine side chain, and histone demethylase for removal of the same. As each enzyme have specific role in chromatin remodeling, they are recruited at specific time. Gene regulatory proteins are initial recruitment signals for these enzymes. In some cases, covalent modifications only exist even after disappearance of a regulatory protein, hence remain as memory developmental history of cells. This figure shows site for covalent modification on a core histones, and there are different modifications are seen in core histones. Histone modification alter nucleosome function in a controlled manner. Covalent modification such as acetylation or phosphorylation will reduce overall charge of histone tails. For example, if you see acetylation of lysine and arginine residues at N terminal increases transcriptional activation of genes. Acetylation at molecular level reduces the number of positive charge at N terminal, which reduces the interaction with negatively charged DNA, and because of this you will find that modification of histones also control interactions with other proteins such as chromodomains and bromodomains responsible for these interactions. Proteins containing bromodomain interact with acylated histone tails, and chromodomain proteins interact with methylated histone tails. Many bromodomain containing proteins may contain histone tail specific acetyl transferase responsible for maintaining acylated chromatin. Similarly, association of tail specific methylating enzymes with chromodomain proteins suggest mechanism for maintaining methylated nucleosomes. Now students, let's summarize what we have learned from this topic. If you see, the structure of DNA is packed in well organized manner so that 6 to 10 micrometer diameter of nucleus is capable of packaging 6 billion base pair of a diploid human cell. Positively charged proteins, which are known as histones, packed together with negatively charged chromosomal DNA into a microscopic space of the eukaryotic cell nucleus. This DNA histone complex we generally call it as a chromatin. Among these, 5 different types of H2A, 2B, 3 and 4 are called core histones, as these proteins are present in core for wrapping of nucleosome DNA. Histone H1 is called as linker histones because it is not a part of core complex. Instead, it binds to the linker DNA between two nucleosomes. Histone H3 and H4 tetramer first bind to the DNA and then bind to two H2A and 2B dimers, lead to the formation of an octamer. Cells maintain structure of chromatin in such a way that all genes will get transcribed into mRNA. For efficient expression of each gene, chromatin dynamic created by covalent modification of histones, which is done by different specific enzyme. Histone acetyl transferase, that's we call the HAT, is responsible for acetylation at lysine, whereas histone deacetylase complexes, that's we call the HDACs, remove acetyl group. Covalent modifications such as acetylation or phosphorylation will reduce overall charge of histone tails. For example, acetylation of lysine and arginine reduces at N-terminal, increases transcriptional activation of genes. Activation at molecular level reduces the number of positive charge at N-terminal, which finally reduces interaction with negatively charged DNA. Thank you.